Welcome to Sekisui XenoTech's webinar, In Vitro Inhibition Studies, Elements of Design, and Important Considerations in Data Analysis. I'm pleased to have Jennifer Horkman here with us today. Jennifer has more than 15 years of experience at Sekisui XenoTech as a senior scientist in our program Oversight department. She serves as scientific reviewer and study director of multiple non-clinical drug interaction-related contract studies across multiple disciplines in compliance with GLP, OECD principles of GLP, and/or Japan MOHW GLP standards. It is my pleasure to present to you Jennifer Horkman.
Thank you, Michael. I appreciate everyone joining us today, and I hope you're all well and staying safe. I'm looking forward to taking this time for a discussion about in vitro inhibition studies. Rather than a traditional outline, I'm planning to discuss many of the frequently asked questions that occur throughout the course of a CYP inhibition study, from protocol design to interpretation of results. And the questions throughout the presentation are not limited to this list. If you attended the virtual SOT meetings, you may be familiar with many of the slides referenced by Dr. Brian Ogilvie, our Vice President of Scientific Consulting here at Sekisui XenoTech. So there are some common terms that are used when discussing why CYP inhibition is important. So cytochromes P450 are the CYPs we'll be discussing, and these are membrane-bound heme-containing enzymes. A perpetrator, or a precipitant, is the drug-causing interaction, and the drug is an inhibitor of drug-metabolizing enzymes. The victim, or object, is the affected drug, and the drug is a substrate of one or more affected enzymes. So the US FDA, along with other regulatory agencies such as the EMA in Europe and PMDA in Japan, recommend evaluating investigational drugs for their potential to be the victim or perpetrator of drug interactions in order to improve the drug's effectiveness and safety. Victim drugs, as we mentioned in the previous slide, are the affected drugs, and this is often because the clearance of the drug is determined by a single route of elimination. And perpetrator drugs possess factors that alter the clearance of a victim drug. Human CYP expression varies enormously from one individual to another, and the causes for variable expression can include genetic polymorphisms. A couple examples would be poor metabolizers or expensive metabolizers of certain CYPs. In addition to genetic polymorphisms, drugs that inhibit, induce, or cause transporter interactions are classified as perpetrators as well. The resulting drug interactions, or DDIs, that occur due to the victim drug can lead to situations where the drug is not approved or even withdrawn from the market. They can also result in loss of efficacy of the drug, leading to labeling restrictions and warnings. When considering the effect of the victim or perpetrator potential, we evaluate the major human xenobiotic metabolizing CYPs. And these are a subset of the 55 functional human CYPs, and they include CYP1A2, 2B6, 2C8, 2C9, 2C19, 2D6, 3A4, and 3A5. These often catalyze the rate-limiting step in the elimination of numerous xenobiotics. They're highly expressed in the liver, although they are present in many tissues. CYP3A4 is highly expressed in the small intestine, and we'll discuss that later. Inhibition can lead to increased exposure in xenobiotics, and a single xenobiotic can inhibit multiple CYPs, potentially leading to large magnitude increases in exposure. The CYP3A4 and 3A5 is also noteworthy that when we evaluate these CYPs, they cannot be distinguished in vitro. A CYP2D6 and 3A4 metabolize about 40% of the drugs and 20% of the natural products in endobiotics. So these are the two most important CYPs for xenobiotic metabolism. So now that we know the major CYPs, what are the types of inhibition? The different types of inhibition can be classified by inhibition caused by the drug itself or inhibition caused by the metabolite or multiple metabolites that are formed. The parent compound is responsible for direct inhibition and time-dependent inhibition. Direct inhibition occurs without pre-incubation, therefore it has a rapid on rate, and time-dependent inhibition occurs upon 30-minute pre-incubation, therefore it's delayed. The metabolite of the parent drug is responsible for metabolism-dependent inhibition, or MDI. This is referred to in the guidances as TDI for time-dependent inhibition, but XenoTech makes the distinction that the time-dependent inhibition that occurs is also NADPH-dependent, as it occurs upon the 30-minute pre-incubation in the presence of NADPH. And we'll go over the different classifications of the MDI in upcoming slides. So direct inhibition is when the inhibitor and substrate bind to the same site on the enzyme. And you can see this in the diagram below. The inhibitor is often a substrate with a higher affinity for the enzyme. This is also referred to as reversible inhibition. And reversible means that the inhibitor is free to come and go from the active site. And the effect decreases as the inhibitor is cleared. In our example, competitive inhibition can be overcome by increasing the substrate concentration. There are several additional mechanisms of direct inhibition, including non-competitive, uncompetitive, and mixed direct inhibition. However, the remaining mechanisms are outside the scope of today's discussion, and competitive inhibition was selected as the most conservative example of the various mechanisms of direct inhibition. For time-dependent inhibition, or TDI, as I mentioned previously, the TDI referenced on this slide is not synonymous with the TDI as referenced in the guidance documents. This inhibition is caused by the test drug after a 30-minute pre-incubation, but does not require NADPH. There is a delayed onset of inhibition, and this is often seen with acidic drugs or oligonucleotides. This TDI can also be due to metabolism by non-NADPH dependent enzymes such as MAO or carboxylesterases, and non-enzymatic degradation can also be a factor. However, we don't know that the TDI we're seeing is clinically relevant due to all of the possible mechanisms that we've provided. The metabolism-dependent inhibition, or MDI, requires NADPH, and this is due to metabolism by CYP or NADPH-dependent enzymes. This is also delayed because metabolites have to be generated first in order to cause the inhibition. An irreversible MDI persists until the new enzyme is synthesized. So just as there are several mechanisms of direct inhibition, I'll be reviewing results and going into greater detail regarding the characterization of the metabolism-dependent inhibition when we review the data. So with all of this information, I have a drug that I want to submit for approval. Where do I start? A typical CYP inhibition study includes the IC50 determination assay, and although the design may appear simple, it must be designed properly in order to not only satisfy the regulatory agencies, but also provide a better understanding of the molecule and prepare for clinical studies. XenoTech CYP design measures direct, time-dependent, and metabolism-dependent inhibition all in one assay. 3 curves will be generated from this experiment, including the 0-minute pre-incubation curve for direct inhibition, the 30-minute pre-incubation without NADPH curve for the TDI, and the 30-minute pre-incubation with NADPH to evaluate MDI. This is a quantitative assay where we determine how much inhibition of the enzyme occurs, and an IC50 value is calculated, which is the concentration of inhibitor causing 50% inhibition of enzyme activity. Some of the controls suggested by the guidance include the no solvent control, and positive control inhibitors for both direct and metabolism-dependent inhibition. 7 concentrations of test article will be evaluated, and the protein concentration, which is human liver microsomes, will be less than or equal to 0.1 mg per mil, and the protein concentration is low to minimize inhibitor depletion and membrane partitioning, otherwise known as binding, and we'll touch on this in upcoming slides as well. Substrate incubation time is short relative to the pre-incubation time. In other words, there's a 5-minute incubation versus a 30-minute pre-incubation, and this ensures that we are only capturing the direct inhibition for the 0-minute pre-incubation samples. This will be performed at 1 substrate concentration at approximately km or equivalent, and along with the short incubation time, conducting the incubation at km keeps the run under initial rate conditions. So this is such that less than 20% of the substrate added is metabolized. During selection of substrates, it's important to note that the FDA frowns upon use of non-specific substrates such as fluorescent probe substrates. The following is a list of commonly used in vitro probe substrates and their marker reactions. And this can be found in Table 1-1 on the FDA's website on drug development and drug interactions, along with a list of suggested positive control inhibitors that's located in Table 1-2. So now that we understand conditions for the IC50 assay itself, how much test material is needed? This is by far number 1 most frequently asked question our protocol design team receives. Another question that often comes next is, why do you need so much test article? My colleague Patrick conducted an ADME 101 detailing the requirements I'll be highlighting here for CYP inhibition, and that recording is a great reference for sending materials for all study types in addition to CYP inhibition. That recording can be found in the Resource Center on our website, along with many other resources, and I'll provided the link here. So the amount of test article required is going to depend on properties of the test article itself, the highest anticipated concentration to be evaluated, and solvent and solubility considerations. The items highlighted on this slide are information we request from sponsors when designing the protocol. The molecules with the larger molecular weight will require more compound to prepare the assigned concentrations required. And the route of administration is important, as regulatory guidance specifies different parameters that are dependent on how the test article is administered. For instance, if the drug is dosed orally, more compound is ingested, and this results in a higher concentration exposure in the gut. The Cmax value indicates the maximum drug concentration in the plasma at steady state and provides information on the anticipated systemic exposure. The plasma free fraction helps identify compounds that are more abundantly protein bound and therefore they have a decreased bioavailability. And the log P partition coefficient helps us understand aqueous properties of the compound. As I touched on briefly in the IC50 assay design, we need to consider nonspecific binding and distinguish between binding to the apparatus and partitioning into the human liver microsomes in the design for these studies. So based on our experience, if human liver microsome concentration is less than 0.1 mg per mil, minor effects are noted if the log P is less than 4. So the concentrations that define the highest incubation concentration to be evaluated, as I mentioned, will depend on how the drug is dosed. So if the drug is dosed orally, this will be applicable for enzymes that are highly expressed in the intestine compared to other tissues in the body, such as CYP3A4/5. So you would use 0.1 times the IGUT, where IGUT is the apparent intestinal luminal concentration, and this is the concentration equal to the maximum dose in 250 ml or a glass of water. So for drugs that are not dosed orally, this is applicable for systemic concentrations or ubiquitously expressed enzymes, such as CYP1A2 or the other non-3A4/5 enzymes. In this case, we would use 50 times the unbound Cmax at steady state, where Cmax is the maximal drug concentration in the plasma. We want to know the unbound concentration because that will give us the worst case scenario. And if the fraction unbound is unknown, a conservative approach is taken where we assume 100% unbound. So once the concentrations are determined, there are several considerations regarding the solvent used to dissolve the test article. Some organic solvents can inhibit or activate enzymes at high concentrations, causing a false result. FDA guidance states that the sponsor should keep organic solvents at low concentrations, less than 1% per volume, and preferably less than 0.5% in the final incubation. For test articles used in CYP inhibition studies, XenoTech limits all organic solvent percentages to 0.2 for solvents such as DMSO to 1% for solvents such as methanol or acetonitrile. And this is, of course, depending on solubility in the test system itself. As we begin to discuss solubility, we sometimes hear, my drug is not very soluble in aqueous solution. Can you test pharmacologically relevant concentrations, and more importantly, will the data mean anything? And the FDA guidance does allow for evaluation at the limit of aqueous solubility. However, this could be time-consuming process and require more material if you don't have solubility information for your compound in aqueous solutions. We will also be asked, can you include BSA to improve solubility? This is not something that we would recommend because compounds that are not soluble in aqueous solutions may be more lipophilic and therefore more likely to bind to proteins. So while BSA may improve the solubility, it comes at the price of a reduction in the free concentration. So now that we have enough materials and conduct the IC50 assay, we get to the fun stuff, and this is the results. What do my data mean? As a guide, this is our CYP inhibition decision tree that helps us navigate next steps depending on what type of inhibition is observed. We will be reviewing data for both direct and metabolism-dependent inhibition, or TDI, is referenced in the guidance document from the one IC50 assay that we perform. This is an example of IC50 results for the evaluation of inhibition of CYP2D6. In this case, direct inhibition was observed as an IC50 value was calculated to be 11 micromolar upon the 0-minute pre-incubation. So 50% inhibition of the enzyme occurs at about 11 micromolar, and your IC50 value is defined below. For the most part, the curves that were pre-incubated were superimposable, as the inhibition did not increase upon pre-incubation with or without NADPH, indicating that there was no time-dependent effect. And we can also see this with the similar IC50 values that were calculated for each of the curves. Therefore, this drug would be a direct inhibitor of CYP2D6. So where do we go from here? We will need to determine a Ki value, which is the affinity of the inhibitor for the enzyme. This Ki value is also required to determine clinical significance of direct inhibition. The FDA and EMA recommend that you calculate the R1 value associated with reversible inhibition. The Ki value required in the FDA guidance basic model can be used to determine the maximum total free systemic inhibitor concentration in plasma that would trigger clinical significance. So if your R1 value is greater than or equal to 1.02, or your R1 gut is greater than or equal to 11, the sponsor should further investigate the DDI potential by either using mechanistic models or conducting a clinical DDI study. So these are our critical values. Under our assay conditions, the Ki value that's used in this equation can be conservatively estimated as IC50 divided by 2. So the question arose, do we really need to perform a Ki determination? Sekisui XenoTech published these data in 2015 with collaboration from Dr. Andrew Parkinson. And data from 343 experimentally determined Ki values over the span of a decade correlate with predicted Ki values where Ki is equal to IC50 divided by 2 when the substrate concentration is equal to Km. So this is another reason why the substrate concentration is critical for this assay to allow these predictions. This was based off the Chang-Krusov equations, and this publication is now cited in the 2020 US FDA final guidance document as an acceptable method for determination of the Ki value. So now that we've determined the clinical significance of direct inhibition, we move on to inhibition caused by a metabolite or potential metabolism-dependent inhibition. In these example data, the test article is a direct inhibitor, and the inhibition does not increase with pre-incubation in the absence of NADPH. And you can see that with this left graph here. However, when you compare that pre-incubation of the pre-incubated samples minus NADPH, there's a 23 micromolar IC50 value. When you compare that to the samples that were pre-incubated with NADPH, the IC50 drops all the way down to 0.23 micromolar, which is a decrease by a factor of 100. The criteria for classification of MDI is cited in the survey of industry referenced below, where a drug is a metabolism-dependent inhibitor. The IC50 value decreases by a factor of 1.5 to 2-fold. And if the IC50 is not established, there is a 20% decrease at any concentration. So when using the IC50 shift approach, a decrease of 1.5 to 2-fold represents a significant enough change to categorize a new molecular entity as a potential TDI. XenoTech takes the most conservative approach to this suggested range, and we classify MDI if there's a shift or a left shift of 1.5-fold. So the different types of MDI include reversible MDI, irreversible MDI, and quasi-irreversible MDI. For reversible MDI, the metabolite is simply a more potent direct-acting inhibitor, or it's reversible. Because the inhibitor is free to leave the active site, it is not actually inactivating the enzyme. And this type of inhibition can be overcome by the use of the saturating concentration of marker substrate, as well as the dilution of the pre-incubated sample. Both are used in the standard design of the Ki and KNACT. Irreversible MDI is where the metabolite covalently binds to the enzyme or active site, so it does not leave the active site and basically binds irreversibly. This can also be referred to as mechanism-based inhibition or a suicide substrate. Quasi-irreversible MDI is where the metabolite coordinates very tightly to the heme iron of the cytochrome P450. In other words, a metabolite inhibitory complex, or an MIC, is formed, and it is essentially irreversible over the time that the enzyme exists. It may be helpful before moving on to the Ki and kinact determination to ensure that the MDI does in fact result in an activation of the enzyme. We conduct this assessment of reversibility using the XenoTech reversibility assay, or the XTRA. This is a qualitative assay that helps us define what type of metabolism-dependent inhibition occurs. One concentration of test article is pre-incubated with human liver microsomes at 0.1 mg per ml, anywhere from 30 to 120 minutes, depending on the results of the IC50 experiment. Unmetabolized test article and reversible inhibitory metabolites are removed from human liver microsomes by ultracentrifugation. There are three typical groups of samples that are prepared prior to measuring residual enzyme activity. Group A includes samples that are pre-incubated only, and this will replicate the IC50 inhibition that you saw in the results. Group B are samples that are pre-incubated and microsomes are re-isolated. And Group C are samples that are pre-incubated and the microsomes are treated with 2 millimolar potassium ferrocyanide prior to centrifugation. And this will dissociate the quasi-irreversible MICs that might be formed. And the marker substrate concentration will be 10 times Km, solubility permitting for the substrate solution itself. For review of the upcoming extra results, each of the bars on the graph represents the treatment groups discussed in my previous slide. In this case, the no re-isolation samples in Group A represent inhibition observed in the IC50 assay. So you can see from the solvent control where the inhibition has occurred. These data were generated upon incubation with the known CYP2C19 reversible metabolism-dependent inhibitor, R-fluoxetine. As you'll see, the MDI was reversed upon centrifugation for re-isolation as the percent of control activity returned to that of the solvent control level. So here, it did have a return to solvent control level. Therefore, unmetabolized test article or reversible inhibitory metabolites were removed from human liver microsomes by ultracentrifugation. So the metabolite is a more potent reversible inhibitor than the parent. For these results, the known CYP2C19 quasi-irreversible metabolism-dependent inhibitor S-fluoxetine did not have the return to percent of control activity just from re-isolation alone. After centrifugation, it just irreversibly persists. However, upon treatment with potassium ferrocyanide prior to re-isolation in Group C, there was a return of the percent of control activity to that of the solvent levels. So the treatment with potassium ferrocyanide dissociated the MIC complex, causing the MDI. In other words, this is a quasi-irreversible inhibition. Finally, the known irreversible metabolism-dependent inhibitor, ticlopidine, inactivated 2C19. This is evident as the centrifugation and treatment with potassium ferrocyanide prior to re-isolation did not overcome the inactivation, and there was not a return of the percent of control activity for either of the treatment groups. Therefore, there is irreversible metabolism-dependent inhibition. Since quasi-irreversible and irreversible MDI result in an activation of the enzyme, regulatory agencies may require you to determine the Ki and kinact values associated with this enzyme inactivation. And this is done with our Ki and kinact assay. So the IC50 is only a snapshot, usually after 30 minutes, whereas the kinact gives a broader picture of what's happening over the entire pre-incubation period. This is a quantitative measurement of MDI, and it includes 5 inhibitor concentrations plus a solvent control, 5 pre-incubation time points plus a 0-minute pre-incubation, and this must be performed at saturating marker substrate concentration, in other words, 10 times Km. This usually incorporates a 10 to 20-fold dilution. We use the dilution here because we know we're inactivating the enzyme, and since the test article is still present in the incubation, we have to dilute the samples to minimize the effect of any direct inhibition. So these are example data from our Ki and kinact assay, and you are provided with an inactivation decay plot and also an inactivation direct plot. And the Ki value that's determined is the concentration of inhibitor that produces half of the maximum rate of inactivation. The Kinact value is the maximum rate of enzyme inactivation. And we also calculate an inactivation efficiency value by Kinact over Ki. As you'll see, when the Kinact inactivation direct plot is compared with the Km and Vmax direct plot for metabolism, Kinact for inactivation is analogous to Vmax for metabolism, and the Ki is analogous to Km for metabolism. So this Kinact and Ki efficiency value is also similar to in vitro clearance of Vmax over Km. Because MDI does result in an activation of the enzyme, metabolism-dependent inhibition is often of greater clinical concern than direct inhibition, and the FDA guidance requires calculation of the R2 value for basic models of TDI. If the R2 value is greater than or equal to 1.25, the sponsor should further investigate the DDI potential by either using mechanistic models or conducting a clinical DDI study. So in conclusion, if you have negative data, you can completely remove the need for clinical DDI studies with the R calculations. Positive data can identify which DDI should be investigated clinically and can assist in the design of the clinical studies. Positive data may potentially restrict co-medications during the clinical trials, but at least you're aware of that moving forward. Although there may be labeling implications as well, overall, evaluation of CYP inhibition improves the safety profile of drugs and other registered xenobiotics. I can't pass up the opportunity to thank several people that contributed to this training. First and foremost, thanks go to our sponsors. It's their collaboration that makes this important work possible. I also want to thank Dr. Brian Ogilvie and our entire scientific staff, as there are countless resources for experts in the field here at Sekisui XenoTech. I'm forever grateful to Lois Haupt for her guidance and management. And finally, I want to thank our marketing department for enabling us with the opportunity to provide this resource. Thank you.
Thank you, Jennifer. And while we wait for questions, I wanted to mention that information about our services and test systems, published papers, posters, other webinars, and more are available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Jennifer's presentation or any of Sekisui XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website. For today's Q&A, Jennifer will be joined by Lois Haupt, a principal scientist in our program Oversight department with 20 years of experience at Sekisui XenoTech. Jennifer and Lois, feel free to start answering questions whenever you are ready.
I may need you to answer or read the question, Lois.
Yep, I sure can. The first question is, in time-dependent inhibition, do you look if only parent drug is present in the incubation, or do you look for metabolites if there are metabolites formed by non-enzymatic degradation or other enzymes? So the answer is, in the CYP inhibition experiments, we're focusing, these experiments have been optimized for our marker substrates. They've not necessarily been optimized to look at any metabolism of the drug that we've added to the incubation. So we typically do not look for any products of the drug itself. We're only focusing on what's happening with the marker substrate that we add to the incubation. So. And then the next question is, do we determine unbound Ki in these experiments? We can. We don't necessarily experimentally determine that, but we provide the Ki value, and then there are options in the guidance to use the log P value that we've requested, or that we typically request, that Jenny pointed out, and we request in our protocol process. We can use that value to calculate the theoretical microsomal protein binding and then correct values for that. So that is an option.
All right. The next one is, in early drug discovery, is it necessary to confirm time-dependent metabolite-based inhibition, or is direct inhibition the only relevant test at that stage?
So in early drug discovery, it depends on how you're going to use the data. So if you're in early drug discovery and you are looking to take those data through the approval process, you will need to look at time-dependent, metabolism-dependent inhibition. It also depends upon how your company screens for drugs. Some of the companies that we work with will actually screen compounds for any metabolism-dependent inhibition liabilities. So it depends on the priorities of your company, but at some point you will have to answer that question, and it's up to your company to determine, do you want to weed out those compounds earlier in the development process, or do you want to take them to a certain point and then figure that out?
I apologize, that was the only question I could see.
Okay. So what is the rationale of using NADPH in the CYP assay? Is it universal for metabolic enzyme, all metabolic enzymes assay? So the reason we use NADPH in the CYP assay is that we're looking at the cytochrome P450 enzymes. We're using human liver microsomes. With human liver microsomes, you have to supplement the cofactors for the enzymes of interest, and NADPH is the cofactor for cytochrome P450 enzymes. So when we do fortify our incubations with NADPH, we are only looking at CYP enzymes. If we were to look at another microsomal enzyme such as UGTs or something like that, we would have to supply the appropriate cofactor for that enzyme. Okay, so is there a way to investigate phase 2 metabolites in an inhibition study? There are ways to do that. If you think that phase 2 metabolism is very important in the metabolism of your drug, and if you think that, so if there's the possibility that using human liver microsomes fortified with NADPH, we may overpredict the inhibitory potential. We could potentially use cryopreserved human hepatocytes. So that is one option. And I see there's another question further down. We do occasionally do in vitro CYP inhibition studies using hepatocytes. However, the hepatocytes is a very complicated test system, and so we are very careful about recommending that. When you're looking, the idea behind the cytochrome P450 assays is that we want to keep the test system, we want to keep the metabolism of the substrates as simple as possible. So we typically recommend the simplest test system first. So we typically recommend human liver microsomes first. If there is a significant contributor of phase 2, and we suspect that system-dependent inhibition may happen, then we may recommend using hepatocytes. There are other cases. For example, we know that oligonucleotides with a certain backbone or certain structure can overprotect CYP. We can overpredict CYP inhibition in microsomes, and we may use hepatocytes in that case as well. So here's a good question. So are all of the in vitro studies conducted before or along the lines of animal testing? So that is a very good question, and honestly, we get CYP inhibition studies for compounds that are in all stages and various stages. So sometimes clients will have compounds, will have done some animal testing on the compounds. Sometimes we have clients that want to just screen a panel of compounds just in vitro before moving anything further along in the animal or in the testing process. So it ultimately depends upon, there's no set recommendation by guidance. It just ultimately depends upon the company's, what the company, you know, do you want to take, if you've got a compound, do you want to take it through all of the animal studies first and then find out that maybe it's a metabolism dependent inhibitor in human, or do you want to do that testing first? So it's something that you have to decide as a company. In an in vivo situation, this is a good question. In an in vivo situation, what is the implications of having irreversible versus quasi-irreversible metabolism dependent inhibition? Is quasi-irreversible inhibition less of a concern? So there really is no difference with what you're going to see in vivo between quasi-irreversible and irreversible inhibition. You're going to see them. It's not like we can dose patients with potassium ferrocyanide and reverse anything. So it's all going to result in inactivation of the enzyme. There is some literature out there that does suggest that maybe irreversible metabolism dependent inhibition could be more of a cause of some of the idiosyncratic drug reactions that we see. But I don't know that there is anything that conclusively has decided that, because with the irreversible metabolism dependent inhibition, you're more likely it's a covalent bond, and so you're kind of modifying the enzyme, the protein itself. So there is that school of thought. But the main reason we run the extra is just to make sure to rule out those compounds that are reversible metabolism dependent inhibitors. Would you please talk about the need and how to determine fraction unbound in microsomes, especially for highly protein-bound drugs? So the FDA does, the most recent guidance does emphasize, you know, making sure that you don't have a highly bound drug in your test system. So there are several ways that you can look at that. You can do calculations that are listed in the guidance using a log P and determine the theoretical fraction unbound under your assay conditions. If you want to determine it experimentally, there's several different processes. If you're familiar with plasma protein binding, the same process is kind of applied. You can do rapid equilibrium dialysis. You could use ultrafiltration. You could use ultracentrifugation. So ultimately, if you're determining the fraction unbound in microsomes, it's going to determine on what it's going to, you know, what process you use is going to be dependent upon what capabilities you have as a company or what information you have available. So the question is, how would Sekisui XenoTech handle a Schedule 1 drug in a CYP inhibition assay? This is an interesting question. I think they're referring to the DEA Schedule 1. We do have a DEA license. We can handle drugs that are Schedule 1 on the DEA, they're Schedule 1 according to the DEA. What we would do is we would handle it just like we would, we have procedures in place. Our safety officer, we have certain individuals, we have a dedicated storage area for those compounds in order to, and we meet all of the requirements of the DEA to handle those kinds of drugs. So other than having the test article locked in a different area, we would store it in a different place. But as far as the assay goes, it doesn't really affect how the assay is run. So if we do not estimate the unbound fraction, can you assume that the Ki could be the Ki unbound as the worst case? That would definitely give you let me think here. So it's I over Ki. Ki unbound may actually underestimate, I think. I think the Ki unbound, because it's in the denominator of the calculation. So a lower Ki concentration is going to make that calculation larger. So it could definitely underestimate your R value. Can you explain the reason why MDI is significant in the presence of NADPH? So the reason why having NADPH is significant when you're looking at MDI, because we're focusing on cytochrome P450 enzymes. When we pre-incubate in the presence of NADPH, if the test article itself is a substrate for cytochrome P450 enzymes, the presence of the NADPH, even though it's only a 30-minute pre-incubation, we may be generating enough of those cytochrome P450 metabolites of the test article to have an impact on our marker substrate metabolism. So it's important to understand how that CYP metabolism, how that drug, when that drug is metabolized by CYP enzymes, it's important to understand whether those metabolites are also inhibitory. So I hope that answers your question. Even if it's not a potent CYP3A4 inhibitor, due to high oral dose level, the calculated effect could be overestimated based on FDA guidance. Can you comment on that? The reason the FDA has put the emphasis on CYP3A4 in the intestine is mainly because of the interactions with grapefruit juice that have been identified over the years. So they found that grapefruit juice was inhibiting the CYP3A4 or CYP3A enzymes in the intestine and causing drug-drug interactions. So that's why they focus on that. Now, all of these in vitro assessments that we're looking at, all of these calculations that Jenny presented are typically conservative approaches, and they do tend to overpredict. And so that's where if something triggers, then you may want to do some, and you don't want to do a clinical study, you may want to look at some of the more sophisticated models and see if once those sophisticated models are applied, does it still trigger does it still trigger for a DDI study? Let's see, do you also look at inhibition study with cofactor, NADPH, and UDPGA? Typically, we only look at one or the other. So we can look at UGT inhibition, but we look at that in a separate experiment than the CYP inhibition experiment. Now, if you have a test article that you suspect is highly metabolized by UGT even before the CYP enzymes get involved, that's where we would recommend looking at maybe a hepatocyte test system or something like that. But we don't typically run these experiments with both, just because, again, the FDA wants you to use substrates that have a simple metabolic scheme for your assays. And if you start introducing UDPGA, then you have to start thinking about contributions by glucuronidation. So does this cover if AO is involved in metabolism-dependent inhibition? We're not really looking at aldehyde oxidase. That is typically a cytosolic enzyme, and so these assays are not cytosolic, I'm sorry. It is a mitochondrial enzyme, and we're looking at human liver microsomes, which is a subcellular fraction. It's actually the the endoplasmic reticulum membranes. And so we don't typically focus on AO. If you wanted to look, if you wanted to look at that, we would have to have separate discussions and design maybe a unique study type for that. Okay, so here is an interesting question. What is your opinion on the cocktail approach for CYP inhibition? So we do offer a cocktail assay as part of our screening, our Discovery Plus screening platform, where we do have a cocktail of CYP1A2, CYP2D6, CYP2B6, CYP2C, all of the major enzymes, except that we only include one of the CYP3A4 enzymes. So that is typically used in a discovery mode. It's not necessarily something that would be, we don't know that the regulatory agencies will accept it, and probably not for the submission for an NDA. The reason being, the FDA requires 2 marker substrates for CYP3A4, 2 structurally unrelated, and you cannot incubate 2 CYP3A4 marker substrates in the same cocktail. So it's good, I would say it's good for a screening approach, but again, you've got all of those substrates there. The FDA prefers to have a simple metabolic scheme. Each of those substrates is going to interact with the other substrate. So for a screening process, it's okay, but when it comes to submission and determination, we typically recommend doing the more individual definitive approach. How do you care for crosstalk during analysis? I'm not sure what this question is asking for, unless they're concerned about interaction of the test article with our marker substrates. We do include several controls within our assays to monitor for test article interference with our assays. All of our analytical methods have been validated in the absence of the test article. So we do include test article interference check samples just to make sure that we're not getting any peaks that's due to the test article itself. We do also include test article suppression check samples, and those are looking to make sure that the increasing concentrations of test article are not suppressing maybe our internal standard or something like that. We also use deuterated analyte as our internal standard. So those are some of the controls that we include in the assays to correct for that. Have you experienced with a phase 2 metabolite, considering this is a major, giving irreversible metabolism-dependent inhibition when tested in the test system? So I don't know, we don't always hear back from our clients when we work on drugs what's happening. We do know we've published extensively on gemfibrozil glucuronide. It is a metabolite of gemfibrozil. It's the glucuronidation product. It is actually a metabolism-dependent inhibition, a metabolism-dependent inhibitor of CYP2C8, and so that is the big example that we've got. And so if you know, or if you suspect that there is a phase 2 metabolite, we have a couple options. We could do, we could use a hepatocyte. We could use hepatocytes as the test system, or we could look at that phase 2 metabolite itself as use that as a test article and look at and see if it's a metabolism-dependent inhibitor. Do you have experience with inhibition studies of macromolecules? We have, you know, as the industry has grown and progressed, we do have clients that come to us with oligonucleotides. We have looked at mRNAs. So we do have some experience with inhibition studies for those macromolecules. It just depends upon what type of molecule it is, because we found, for example, I mentioned earlier, oligonucleotides with a certain backbone are time-dependent inhibitors in microsomes, but they are not. But we suspect that microsomes may be over-predicting because there haven't been any clinical any clinical studies that we're aware of that have suggested that they could truly be metabolism dependent inhibitors. So depending on the type of macromolecule, we may recommend using a different test system than microsomes. So, but we do have some experience with that, and we have several publications on oligonucleotides that you can access from our website. In the TDI shift assay, what fold IC50 shift do you consider as a risk for TDI based on clinical DDI? And I think Jenny briefly touched on this. We follow the recommendations of the Grimm et al. White paper that was cited in the slides. They recommend using a 1.5-fold to 2-fold shift in IC50 value. And so we actually use the more conservative 1.5-fold shift. Okay, and then the next question is, for CYP3A4, do you still need to look at testosterone and midazolam as substrates? So the FDA guidance does require looking at 2 structurally unrelated marker substrates for CYP3A4. So yes, that is definitely still part of the requirements for the regulatory guidance. And then we have a question, does the 1.5-fold IC50 shift apply to all CYPs? And that is yes, it does apply to all of the CYP enzymes that we study. So the next question is, how can I select the substrates required concentration for a substrate metabolic enzyme test? For example, I'm using a hepatic cytosolic fraction from XenoTech to check NAT1 activity for a substrate. How can I select what amount of substrate concentration is required? So I'm not 100% clear if this is referring to an inhibition assay or if this is just looking at the activity. If you're designing an inhibition assay, you generally want to be performing these assays under initial rate conditions. So we usually recommend performing the assay at the KM associated with that particular marker substrate. Some of our test systems, I know our pool of 200 microsomes, we do provide that information for you. Some of the other test systems, we don't, it's not included on the product sheet, so that may have to be determined in your laboratory. For the extra assay, did we normalize the liver microsomal protein concentration after centrifugation? So that's a good question. We do actually normalize that. It is important to normalize that protein concentration so that you're comparing everything with the same rates. Also, what if the metabolite is highly bound to liver microsomes, which could not be removed by ultracentrifugation? So if the metabolite is highly bound to the human liver microsomes and it's not reversible after ultracentrifugation, then that's where we're doing the testing to figure out, is it reversed by potassium ferrocyanide? If it's not, then we consider it to it's consistent. The inhibition that we're seeing is consistent with irreversible metabolism dependent inhibition. If it is reversed, then that's where we consider it to be quasi-irreversible. Regardless, if it's not reversed by ultracentrifugation, then it's representative of an activation of the enzyme. And so you would need to determine how quickly that inactivation occurs using the KNACT assay. So the next question is asking the difference between metabolism-dependent inhibition and mechanism-based inhibition. And that's a good question, another good question. Mechanism-based inhibition is a type of metabolism-dependent inhibition. So mechanism inhibition is more consistent with the quasi-irreversible and irreversible metabolism-dependent inhibition. It involves the formation of a metabolite that is not being released. And so that process is inactivating the enzyme. Now, you can have metabolism-dependent inhibition that's not mechanism-based. And that's where, in the slides where Jenny was showing, that's the case of the reversible metabolism-dependent inhibition. So you have a metabolite that is forming that is being released from the active site. It's free to come and go. And so you're not actually inactivating the enzyme. Okay, so the next question is, would you expect the Ki and Knet values to be similar when determined using the dilution versus non-dilution approach? You have to be very careful about using the non-dilution approach. The main reason is you have to, if you're not reisolating those enzymes, if you're not reisolating the microsomes, then the test article is still present. Anything that's unmetabolized of that test article is present, and so you could have some direct inhibition. So you wouldn't want to use the non-dilution approach if you have a really potent direct inhibitor. If using the saturating concentration of substrate is enough to overcome that, then you could potentially use the non-dilution approach. But it can be difficult to tell that. And that's why we also include the 0-minute pre-incubation when we do design those assays, is because that tells us, those samples can tell us whether we are between the dilution, between the marker substrate concentration, are we counteracting that direct inhibition caused by the unmetabolized test article that's still present? The next question is, what's the best way to estimate the highest concentration in research where clinical Cmax is not available? That's a good question. There's really not a way for us to estimate that highest concentration unless you know, potentially, if you have an idea of the dosage, we could use that to design concentrations based on those dosages. If you absolutely don't have any information, I mean, I guess the best approach, the FDA does recommend testing as high as is reasonably soluble. So that could be an approach that you could take. But just know that if we identify it as an inhibitor, you may have additional steps that you have to take to determine if that inhibition is clinically relevant once you have the appropriate data. So the next question is, what if the perpetrator is an enzyme inducer? Why concentrate only on inhibitors? So that's another good question. So we don't focus on enzyme induction for CYP inhibition assays. The reason being, we're using a subcellular fraction, human liver microsomes. Also, our treatment period, the microsomes are only treated for a very short period of time. I mean, we're talking a 5-minute marker substrate incubation and a 30-minute pre-incubation. So generally, that's not enough time to experience induction, because induction involves the, it's a little bit more complicated than that. Now, we have had some presentations that focused on enzyme induction, and those, I believe, are available on our website. So the next question, related to index substrate, FDA DDI guidelines says clearance of bupropion is no longer a CYP2B6 specific substrate and recommends the ratio of hydroxybupropion to bupropion? Do we plan to measure the ratio in your experiment in XenoTech? So that's another good question. We actually have switched to efavirenz as our standard CYP2B6 marker substrate. We found that the Km is lower for efavirenz, therefore it has a higher affinity for CYP2B6. We only run bupropion, the bupropion assay. It's still available. We had validated it and everything. We only run that on a case-by-case basis. If there's some reason the test article might interfere with the efavirenz assay, or if we're using a test system like hepatocytes. So we don't generally run bupropion or recommend it for the time being for as a CYP2B6 substrate. So the next question is, can you make a direct comparison in IC50s with reversible inhibition and TDI? So, so for the IC50s, and I don't know if this is reversible direct inhibition or reversible metabolism dependent inhibition, but the IC50 for the reversible direct inhibition is you can predict the Ki value as half of the IC50 value. If this is talking about reversible metabolism dependent inhibition, so we can look at the plus minus, the IC50 shift with the plus minus samples to see if that triggers for metabolism dependent inhibition. Now, if like in the example that we had in the data, if we find that the IC50 value is a little bit higher after that 30-minute pre-incubation minus NADPH, we could compare. The reason why we can compare back to that direct inhibition, the 0-minute pre-incubation, is we're making the assumption that the test article is not being metabolized. So any inhibition that's happening after that 30-minute pre-incubation would be due to the test article itself. And so that's kind of how the comparison to the 0-minute incubation is rationalized. So the next question is, what if solubility limits the concentration up to 0.1 times the IGUT or 50 times Cmax? And that's kind of what Jenny was discussing on the solubility, the slides addressing the solubility. Basically, the FDA is very clear that you can only test inhibition up to whatever is actually soluble in your system. And so that's where we would test as high as we reasonably could, as high as solubility allows in the test system. And that's how we would, those are the concentrations that we would use in the assay. So that's what we would recommend there. And the FDA is very clear that you cannot test concentrations that are not soluble. So for the KINACT and KI study design, what is your criteria for choosing the highest concentration tested? We don't necessarily have criteria for the highest concentration tested. What we're looking at, so we're looking at the IC50, shift and trying to determine what concentration range are we seeing the metabolism dependent inhibition, and that is the range that we focused. Sometimes we won't go as high as we tested in the IC50 because if there's a lot of direct inhibition, then we may not want to include that concentration, because again, when we run the KINACT assay, we're going to have the test article, unmetabolized test article, present in the incubations. So it's looking at where does that metabolism dependent inhibition start. So the next question is, do you consider necessary the assay to determine the Ki of the inhibitor even if the prediction of Ki equals IC50 divided by 2 is valid? So the FDA said it's not actually necessary because you can use this prediction. Now it's going to determine whether the Ki assay is necessary. It determines on the budget that you have Do you have money that you can spend on this experiment when you can use the prediction to meet regulatory guidance? We do have clients that do automatically, if there's an IC50 calculated, we do automatically proceed with the KI determination to determine the mechanism of the inhibition in the affinity. So it ultimately is going to be a decision that you have to make as a company, whether that's necessary or not. So can you highlight how will the data be used to design a clinical DDI study? That's where the R1, R2 values come in. That's going to be information that's helpful to design the clinical study. There's also software out there that you can plug the in vitro numbers that we determine in and help you see kind of what concentrations, help you design that DDI study. So all we can tell you is if a study may be necessary, it's not, we can't really help to design the actual clinical study itself. So should you perform TDI assays with or without dilution? So this is another good question, and we actually have a whole publication on that discusses when to use the dilution, when not to use the dilution method for TDI assays. If you're doing an IC50 shift experiment, our recommendation is that you do not use a dilution method, and that's because if you are using a dilution process, a dilution method, you may miss that any metabolism-dependent inhibition caused by a reversible metabolite. Our thought is that you should only use the dilution method once you know that you do have metabolism-dependent inhibition and you know that you're inactivating the enzyme. That's it in a nutshell. So the next question is, why are KM values different from laboratory to laboratory? And that's another good question. I suspect that some of those have to do with just differences in how the labs function or slight differences in how the assay was designed. In theory, they shouldn't change, but we do know, and I've actually seen this in our laboratory, if you're changing your microsomal, if you're using different concentrations of microsomal protein, we have seen differences in the KM values. I suspect that may be due to some microsomal protein binding, but that's just my suspicion. I don't have anything necessarily to back that up. Okay, so what is the concordance between IC50 shift and TDI? Do you see an IC50 negative shift for compounds? Okay, so the correlation between IC50 shift and TDI is just basically the IC50 shift is used to determine if we have time-dependent inhibition, metabolism-dependent inhibition happening. In order to know if there's any clinical significance of that, then you need to move forward with the follow-up experiments. And if there's an activation, then you need to determine those K-inact values. If there is a reversible metabolism if there's reversible metabolism dependent inhibition, if you happen to know what that metabolite is that's causing that, and if it is a major metabolite as defined by the FDA, then you may need to design in vitro assays evaluating it as a time-dependent, direct, and metabolism-dependent inhibitor. So if a compound is both an inducer and inhibitor for the same CYP, how do you design the in vitro study? So the studies are completely different. So inhibition assays are run using human liver microsomes, which is a subcellular fraction. We keep things short. particularly for the direct inhibition portion, and we do the 30-minute pre-incubation that's required by regulatory guidance. Induction is a different study type. You're actually taking cultured human hepatocytes. You're treating them for 3 days with the compound, and then you're measuring the mRNA response, or if you want to look at activity, you can look at activity. But regulatory guidance focuses on looking at the mRNA. So it's 2 completely different assays. And so you really can't compare the data. Now, if you're looking at activity-based induction assays, and you know that a compound is a metabolism-dependent inhibitor, you may see some evidence of that metabolism-dependent inhibition over that 3-day treatment in the induction study, but you're not going to see any evidence of induction in the CYP inhibition study.
Okay, it looks like this is going to wrap up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Jennifer for that great presentation, and Lois for her assistance with the Q&A. Have a great rest of your day.